So thank you everyone for joining and welcome to our last session for the workshop. But as we're going to continue further as well. So we'll keep meeting you. So it's technically not the last session. Today we thought we will keep it more as an open table discussion. I just quickly wanted to go through sort of the pipeline of how we have been doing data collection and just show one example of what I and Iqbal did for this month in Patterson Park, which is close to our home over here. So let me share my slide and let me know when you can see it. Can you all see my slides? Okay, cool. So the thing we are going to discuss today is first going to be the data uploading and documentation. And please let us know if you have tried already and you have any issue with data uploading on the website. I will quickly go through it. The other we are going to do is the discussions on the fieldwork. So if you have already started and if you have faced any problems, we will troubleshoot it and address any concerns. And then we will discuss some of the future steps of the program. So just going through the data collection, which I and Iqbal did this month, and we went to the Patterson Park, Lake Baltimore. We talked a little bit about it in the last session. So I'm just following the protocol we sent you all to go step by step on how we are documenting. And the way you want to document is really up to you. If you want to use like a Word document or something to do it, at that time, or if you just want to write it down on a page or just like print out the protocol we sent you and then just tick mark things and write next to it, that's also possible. So another first thing we did once we went to the water body was took a picture of the water body, because that itself gives a lot of information. So if you can see here, it's already very greenish. There's like plant-like stuff floating on the top. So that is sort of one major data point you would have, because imagine if you are in a place where it gets cold over time or it snows over time, or somehow this thing starts degrading and becoming more brown in color, you will be able to see that just with the picture. So the next thing I reported was what is the appearance of the water body. And as I said, it looked like small floating plant-like structure on the top of the water. So very general, whether, you see plant-like or something filamentous-like or soup-like or something of that manner. So we gave you some options in the protocol, but you're free to also describe it yourself, how you see it. The third, we just sort of sniffed the water. So you don't have to like go too close or anything, when you're just walking around the water body, you have some sort of specific odor of that water. And that can also tell you whether there is something probably degrading in it, or often if you feel like it is close to some sort of industrial area or something, and there is, they're releasing something, it can have a very different smell altogether. So when we went to this water body, we felt like it was some sort of a rotting smell. So I wrote down that as my data point as well. Oops, sorry, I don't know why I have this repeated. The next thing I did was to just look at the color of the water collected. So once we collected the water in a jar, you can already see it's slightly greenish in color, and that is also visible from the picture of the water body. So you can just say, okay, this is sort of greenish. We did one more, which sort of looked like a little brownish and reddish because it had more red algae. So you can just report that as well. And here you can see I dipped a pH paper inside and then just reported what my pH of the water sample, which is around 8 in this case, and then I just reported that as well. The next exciting thing, once you have the data, once you have your water sample back at home, is to do full scope and look at the spectrophotometer and check, how the algal growth looks like over months. So the first thing was, again, algal identification using full scope. And you can see in just one sample, we got a bunch of different kinds of algae, and it was very exciting because if you look at the algae over here, I identified it to be oscillatoria. So I used actually the algal web and went step by step, first by saying that, okay, this is a filamentous algae, and then thinking about how it looks like. So it looked like band-like. So I looked for that in the algal web. And if you have any issues using algal web, also please let us know. So we identified it as to be oscillatoria, and you can see they're sort of gliding on top of each other. So also I saw this sort of brownish algae, which I haven't identified yet. So this is something which I can clearly see, but I haven't identified. The other kind of algae, which if you see on the picture on the right, also it's in a form of a colony, I could see was these round algae. And I sort of put it on Twitter and asked people, what do you think it is? Because it was hard for me to sort of figure it out. And some of the people actually made an observation that there is some sort of a membrane sheet around it. So we identified it to be Gliocystis vesiculosa, which I then went around to read more about it because I had no idea about this algae at all. And then I also see some euglena. You can see like a filamentous euglena moving around. And the other one you see moving like in a rounding motion. I checked on the internet, it says it might be a resting euglena, even though it doesn't look like it's resting a lot. It's just sort of in this round shape and sort of moving around. The other colony I see, which I haven't identified yet, is this algae. So I just have it there saying that, okay, it's not identified yet. So you might also get sort of data where if you are not able to identify what kind of algae it is, just you keep that data because then we can ask the experts to identify that algae for us. Okay, so the other data collection, which we discussed last time, is from the spectrophotometer, and we went in detail about it, but just quickly to show you how my data table looked like is just I put all the values which I get from the water and then from the park sample which I got, or the lake sample which I got, and change the paper color, red, green, and blue, and then just report the values I get on my app there by measuring the content, sorry, the algal content in the tube over there. So there's the R value, the G value, and the P value here. And again, you can use some sort of, like a paper or a sheet if you're quickly writing this. Just make sure that you write the sample name and the paper color, or you can again print out the protocol we sent you and just note down the values over there. Now I will go quickly to the website where we are uploading everything, and it'll be nice if anybody has any issues doing that, please let us know, because we are also trying to modify the website and we want to make it as easy as possible for everybody to upload data. Okay, so let me stop sharing here. Are there any questions for me? No? Okay, so going to the website. Let me find that out. Sorry, it's taking a second. Okay. Can you see the website now?
Yes.
Okay, cool. So this is the account I made, and we talked about it a bit in the beginning where, you can request us to make an account, and that will be your own account to sort of make your database. And then we will also have that data on the background, because then we can accumulate everybody's data and create a bigger database. So this is sort of my, oops, it logged me out for some reason. Okay, I'm going to log in. Give me a minute. Okay, it did log me back again. So if you look at the first thing, which is the water body database, again, is generally about, you characterize what water location you're going to, report the algae appearance, algae odor, color of the water sample, pH, all the things I just discussed step by step. And you see all of those values are over here. Similarly, we have a spectroscopy database where, again, you have to make sure that you're choosing the color of the paper. You just chose the sample type, whether you're doing the water or the water plus sample thing, and then report all the values. If you look at here, the table I showed you is pretty much the same table over here. So it gets nicely reported. The nice thing would be now, again, when I go another month over there, there will be with data, all these values, and I can see the change in the algal bloom with that. The third thing, which is really my favorite because I love images, is uploading the images I showed you all. So it makes like a nice gallery over here once you upload each of your images, and you can go more in detail as well. So for example, I sort of wrote small notes about, how the stuff looked like there was some small floating plant-like thing on the top. And then maybe small things about things you see in your movie. So I made like a small GIF over here. Again, the oscillatoria thing I showed you, and then a few of the images of, stuff which I haven't identified yet. So that's all sort of from, thinking about how we want to upload all the data over here. If you have any issues uploading it yourself, you can also just send us an email and we can try to do it for you. But we really encourage you to sort of maintain this month to month, because in the end, you're going to have data from, few months you will be doing these experiments. Okay, so next we wanted to do some sort of troubleshooting because we wanted to sort of open the platform and ask you all whether, you have tried this already and if you have any issues. So I'm going to just stop sharing, and Iqbal, you can go ahead and take this.
Sure. So hello, everyone. In the last session, we talked about spectrophotometry and the basics, and I also showed you how I did an experiment with the tool. So did anyone get a chance to use the device, or just like if you have any questions, please feel free to unmute and let us know.
So Sumati has a question. Do we need to take pictures of Foldscope every month along with the spectrophotometer reading?
Yes, I think there is a bit of confusion about how the project is going to unfold in the next couple of months. So as you probably already know, when you signed up for this program, we have a workshop that we planned initially to train you all into using the devices. And the devices are primarily a microscope, Foldscope, and a spectrophotometer, or you can simply call it a spectroscope. Okay. So now I think we are at this stage where we have essentially trained you all into using this device, right? So you know how to use them. If you have any questions, we are always available to personally sit with you or your students and troubleshoot any problems. Now the exciting part of the project begins, which is a long-term algal bloom monitoring project, where what we are trying to do is map out the algal species geographically in the locations that we have identified or you have identified, and also measure the algal bloom as time goes by. Now the question is, how frequently do we measure, or how frequently do we do an experiment? Now the workshop was kind of intense, that we asked you to wake up every Saturday morning and sit with us and do microscopy. Now it's going to be a little much more laid back, in the sense that you don't have to do an experiment every week or every day. All you have to do is find a water body and visit that water body once a month. You can always do it more than 4 times a month or twice a month, but the minimum is once a month. So if you go there once a month, collect a jar or a bottle of water like that, and that's enough. And then whether you want to do the experiment 1 hour later or maybe 5 hours later with your students, that's totally okay. No problem with that, right? But we sort of request you to do an experiment once a month after you have identified the water body. And the experiment entails doing microscopy and spectroscopy. That is following the protocol, the 2-page protocol that we have shared. That's it. So if you do that, you get all the numbers, all the interesting images that you can find in your sample, and then you just upload it. The uploading part also doesn't have to happen the same day you do experiment. If you feel like you have spent some time and you are busy, you can upload it, at night, whenever you are free, right? So we do not have any rules basically. Our objective is to maximize curiosity, okay, and engage everyone. And some of you are lucky because you have students and friends, so you want to do it in a group. Others probably would do it individually and then post it to rest of us over Twitter or on the website, okay? So yeah, to answer that question specifically, yes, we expect you to use Foldscope, take images of the sample that you have collected, and also do spectrophotometry. If you go back to individual classes or the videos of individual classes, you will find out how to do each one of them, okay? And On top of that, if you have any questions or if you want us to do a separate session for you or your students together, we are definitely going to do that. So no questions asked. Okay. All right. So are there more questions? I'm going to transition into another part of today's presentation. Okay. So we sent a form out to all of you to requesting you to fill it up with the names of water bodies that you have identified, and when do you plan to start, right? So what I'm going to do, and a lot of you have already filled it up. So what we are going to do is we'll call upon you now and just unmute yourself and say a word or two about the water body that you have thought or identified and why you are excited about that water body, or have you ever been there, or how did you hear about it? Just a single line is enough. So what I'll do right now is I'll go into gallery mode, and there is nothing to fear. If you have not identified it yet, it's okay, no problem. Okay, so what we will do is just we wanted to know individually how do you feel about the water body that you have thought of. So on my screen, since Ankita and I have already discussed about Patterson Park water body and given you the details, so I'll start off with Kanika. So Kanika, could you please unmute yourself and tell us about the water body that you are planning to visit?
Yeah, so the water body that I have identified, it is called Lake Merced, which is in San Francisco. So it's a little bit away from San Francisco. The only thing I'm afraid is, like, last time when I went, I think I saw like a sign there that the algal is blooming there, but I think it is now winters, so that might be a good time, and it might be actually good to see the progression, like if next year they again put up a sign, that the algal is blooming there, so just stay away or something. So that have to be careful about. But yeah, otherwise I will be going to Lake Merced, and I think probably should be a good time now to start and then see the progression over summers, how it happens. Yeah.
Wonderful. Yeah. So, I mean, the idea is to monitor bloom. So if you can start monitoring, so by the time summer arrives or maybe early spring, you will see something very interesting. Next is Nikhil's.
Can you hear me?
Yeah, go ahead.
So I'm planning on going to a recreational body of water called Shadow Cliffs.
Mhm.
It's probably within 5 miles of where I live, so I have quick and easy access to it. I've been to it a handful of times, and I remember seeing something resembling an algal bloom a few years back. So I'm interested to see what it looks like now.
Yeah, that's great. And we'll talk more about your project again, Nikhil. Yeah. Next is Dr. Vasavi Datar.
Yeah, hello everyone. Actually, I have visited one lake that I have.
Vasavi, can you speak up a little louder? I'm sorry, I cannot hear you.
Yeah,
Just a little
can you
louder.
hear now? Can you hear
Yes,
now?
go ahead, please.
Yeah, thank you. So I have listed 2 water bodies in the Google form today. I have visited only one of it. Other one I'm planning to visit. And this one, I'm just thinking how to collect the sample, because there is a lot of fence like bushes are there in the shore. So I'm thinking how to, we didn't go collect the samples yet actually. We'll go with that the next month.
Okay, great. Yeah, I mean, I personally talked with Dr. Datar, and we sort of are very excited about collecting the data and maybe see where it goes. Next, if I am right on my screen, is Dr. K. Gunashweta.
Yeah. Hi, everyone.
Hi.
Yeah, well, I have actually planned to take the samples of 2 water bodies from my own hometown. That is one from Lake Mancherial and one from Lake, I think it's called Penganga. So apart from these both, there is one lake nearby our college where I am trying to take my students out and take sample with them. So I think for now, my hometown is like, for to be a safe side, I'm taking about the hometown lakes, 2 lakes. If it's possible to take the students out, then I will go for the third one. So I am just trying to upload the details about these 3 lakes for the our project.
Yeah, and have you tried to make an account on the website so that you could upload data when you have them?
Still not Iqbal. I still have to do that because of this festival holidays they are going
Oh
on.
yeah, yeah, yeah.
So we are a bit busy
No,
in
yeah,
traveling here
yeah.
and there. So yeah, I will
No,
be
yeah,
doing it next weekend.
sure, yeah,
Till
take
next
your time.
weekend, surely.
Yeah,
Yeah.
no, I mean, we are already grateful that during this festive season you took time out for joining the program. Thanks a lot.
Actually, I almost forgot about the meeting. Just a mail has been popped up in the phone, and I just quickly got alerted and joined the meet.
Yeah, yeah. For everyone else who is not in India, this is the time in India when there is a lot of festivals going on across all the communities and all the cultures in India. So it's a busy time. Okay, next
Yeah.
is Sandhya Rani.
Hello everyone.
Hi.
Hi. I've identified a lake near to Ichoda, which is in Adilabad district. The lake is surrounded with mountains, and most of the water collected in that lake is streamed from those mountains.
Mhm.
And the water is used for the agriculture purpose. And I think some water would also be flowed from the agricultural fields. So everything is collected in that lake.
Mhm.
It's a bit difficult to reach that lake, but that's the only one which is near to my college. And I mean, I'm staying at Ichoda. My college is at Ichoda. So I hope I'll find it useful. And I
Yeah,
have one more doubt.
yeah.
Um, I see so many lakes near to my house, but most of them, in most of those lakes, the sewage water is draining in. So obviously, if the sewage comes in, the algal bloom will be so high, right?
Yeah,
So I saw those lakes. I mean, I think 1 or 2 lakes in my surroundings,
yeah.
but mostly it is, collected with sewage.
Mm.
Though it's a fresh water, I mean, it's all rainwater, but sewage is also draining in.
Right.
Can we collect the sample from those kind of lakes?
Well, I mean, I would let others comment. In my mind, if I were you, I would try to go to a water body where not only there is fresh water, but also people use it for multiple purposes like drinking or agriculture or something else, right? So that way, you get information about the water body, which actually is interesting because you would know something about public health and all. But if you're curious, you can always go to any water body and see what is in there. That will also be a data point because you can always compare at least once, what is the difference between these two lakes. In one lake, you have water coming from the mountains. In the other lake, you have dirty water coming in from different sources, right? So that
Yeah.
would be a good comparison. But at the end of the day, you decide what you want to do, which one is convenient, and also which one you think is much more meaningful in terms of collecting long-term data.
Yeah, but and one more doubt is we have to collect the data for 12 months, right? So we have to do that from a single lake, right?
Oh yeah, yes. I'm really happy that you asked this question. So it's very important to continue measuring from the same lake, not
Yeah.
switch lakes in the middle. I mean, you can do 2 lakes and eventually decide on doing 1 lake because it is convenient for you, right? But if you switch lakes, then it's a big problem. Problem in the sense that then it's very hard to monitor, right? What's going on in that lake versus that lake.
Yeah.
Because
Yeah, I have chosen the hometown lakes because of this reason, because it is the house that I can go like per month for 2 days and I can collect this sample. It's like it's +1 for me
yeah,
for all this
yeah.
thing.
Yeah. Some of you like have groups like Pandyarajan and Inian. I know they work in a group, so they can always cover for one another if one is not interested in going that month. Yeah. But if you make it fun, like if you collect your friends and say, let's go and do a small field trip and sit together, have lunch and do some experiments, it's really fun actually. Yeah.
Yeah, that's what me and Iqbal are trying to do as well.
So we meet once a month for lunch and then we do a quick experiment, even in the field, or we bring home the samples and do it there. So yeah, once you do it, it's just the first rate limiting step, right? The first step. So once you do it, it's going to be really fun. Okay, we are running out of time, so we'll quickly move to Dr. D. Snehalata Dodipati. Can you hear me? We can't hear you, so we'll let you fix your audio, and meanwhile I'll move. Oh, we can hear you now. Wonderful.
Actually, I chosen 2 freshwater lakes in our town city. So the to college, but I spend in a home. I will collect the samples. So actually, these are 2 lakes are freshwater lakes. I want to monitor how much of pollutants form in that lake. So I want to measure that water body conditions. Yeah. And one more thing, I try to log in as a member in our Open Field Collective web page.
Yeah.
So it's not that.
Sorry, I didn't understand. You tried to log in and then what happened?
But it can't take that.
Oh.
Actually, it's your email and password is incorrect. I face
Okay.
that problem.
Yeah. Okay. No problem.
I
Yeah.
try to solve.
Yeah, we will. One of us will reach out to you and solve that problem. Does that sound good?
Yeah. Okay. Thank you.
Okay. So if you can stay for like 5 more minutes after this meeting, then we'll try to solve it today itself. Okay.
Okay.
Yeah. Next is Owen. Hey, Owen. Owen, are you there?
Yes,
Okay.
I am planning on testing my pond in Cowan Lake. They're both around my house, so that shouldn't be too long to get each month.
Yeah. So yeah, we saw the picture of the pond that you posted, and Kanika especially was really concerned about you and when and how you would go there.
Because I had been reading about, the algal blooms in Ohio, which are like a complete menace. So I just want you to be careful when you go, maybe go with a guardian and everything. So just have all the safety precautions and that should be fine. Yeah.
Yeah.
Okay.
Yeah, we are really excited what you find out, Owen, and let us know.
Yeah, because Ohio is like, right there where people are trying to work on algal bloom solutions as well, because that's the place where they have identified a lot of algal bloom as well. So it'll be very interesting to see what kind of data you get. Yeah.
Yeah. Okay. Next is Shivalata Kota.
Hello.
Hi.
Hi, everyone. This is Shivalila.
Oh, I'm sorry, Shivalila. Yeah, sorry.
Okay. I identified 2 lakes. One is from my hometown, that is at Bibi Nagar, and another is near Mancherial. But I collected sample from Bibi Nagar only. I will go with students near Mancherial Lake, and I will do that.
So do you already have access to students? Like, have the school?
Yes. They are coming to college now. They are having exams. After completing their exams, I will take them.
That's wonderful. Yeah. Okay. Next is Lydia. Hi, Lydia.
Hi, how are you?
I'm good. How are you?
I'm good, thank you. So the lake or pond areas that I'm sampling are in the town where I teach school. I'm a middle school science teacher. We have one pond that's directly across from our school. It's a public pond that's the water supply is fed in from residential areas. And we have another the community has a lot of lakes actually that the students and families use for swimming and things like that in the summertime. So we're also going to use one of the other lakes that a number of my students have their boats and things like that at. So I'm very excited for it. I haven't done any sampling yet. My first classes will go out this week, pond area across the street from us, and then I'll have to take the samples from that other lake in town.
Yeah. Let us know when you sample and if you need any help. And it would be really exciting, and we would be interested to see your students like collect samples or do an experiment, right? So yeah, I mean, when you please try to make an account as soon as possible on the website so that uploading the data is easier. And this is just for everyone else here. Is that if you have issues with the way we have shown you to upload data, you can always upload data in a different format, like share your Google Drive or something like that, right? So let us know what is more convenient for you. So the idea is to have the data as nicely and organized as possible, right? Yeah.
Iqbal, I have a question.
Yep.
The last time that pictures I have posted in the meeting, those were the pictures just for sampling, that they were not from my hometown lakes. So is it okay for if I upload those pictures in the website in my account, or should I only restrict my photos to only the lakes that I have chosen?
So, so when you keep uploading data from the lakes that you have chosen, it will automatically make a repository there. But for now, in the last 1 month, we all went to different lakes and we looked at onion peels, sugar, and everything that we could find or get our hands on, right? So you can upload anything that you would like, and you'd get a nice gallery on the website.
Yeah, like I just recommend that there's like a image title and an image description. Just put there what you're seeing so that like it's sorting for you is also easier.
Yeah.
So that way you can upload anything. Also, there's like a description.
Also, I think to upload every image, like there is a field which says which lake or water body this image is of. So if you enter the right name, I think all the images will be sorted properly and we'll get the data properly. So you can upload anything that you want, any image.
Yeah, I mean, it is really important to note that you add your name every time correctly. You add your water body name also correctly. So that way, the system will automatically sort which images came from which water body and when, right? Because you added the time, or sorry, the date and the water body and your name and email. So everything will be connected. So you don't have to worry about, about adding information that you did it out of curiosity and not part of your experiment from a specific lake. You can just upload something that you did today, okay, let me see. Can we go to?
Can we have some way of like marking it on the website, just because then after that at the back end we will do, like our analysis of where the algae are. So like, I mean, of course, people are free to upload anything, but is there a way like then we can have some sort of a marker that this is like the algae or this is not? I mean,
as far as it's a location and description, it should be fine.
Yeah,
Sorry,
because we'll have spectroscopy data and everything for a particular location, so we can sort that easily.
Yeah. All right. Next is Inian 46. Love your profile picture. Hi, Inian. You there? Okay, we'll give him some time to fix his audio. Can we go to Sumati Natarajan? Hello, are you there?
Yeah, hello, hi.
Hi.
Yeah, so I'm planning on 2 ponds actually, one on the way to college and one nearby to my home. So maybe the one on the way to college can be, I can take the students' help. So once they are back after vacation to college, and yeah, I plan to use these two for the blue monitoring.
Okay, yeah, so let us know when you want to start the experiments. You can start it when the students are back. You can also start it when if whenever you want. So the idea would be to continue doing the experiment for at least 6-7 months, right? And we'll talk about the documentation or publication opportunities that we will have at the end of this year. So there is one kind of documentation that we'll obviously do with National Geographic, and there can be other kind of popular or scientific journals where we add our data and publish our findings. So you all will be part of that because you all have easily identified field or water bodies and you're going to take sample. Just make sure that you take it responsibly and repeatedly from the same. That way we'll get a trend from that water body. Okay, moving on, we go to Kirti Bitlingu.
Hello, hi.
Hi.
I have chosen 2 ponds and which are very near to my hometown, that is Adilabad. One is Satnala and another one is Bangarguda. And actually, the main reason for choosing these ponds is at the time of my degree days, means at that time we had botanical tour. So at that time we collected algae from that project, that is a Satnala project. Actually, it is a tourist area. So again, I'm taking that opportunity to take samples from that pond.
Yeah,
and that is one thing. And another thing is that most of the water is useful for agriculture purpose only. And a few algal members are very useful for agriculture, and what kind of algae is existing in that water, I want to know. So that is the reason.
That's great. Thank you, Kirti. We'll go to A. Santosh. Hello, hi, A. Santosh, are you there? Okay, we'll move on to Vijaya Lakshmi.
Hello, good evening. Good evening, sir.
Hi.
Hello, hi. Due to some personal reasons, I couldn't get all those algae and all those, sir. And the next week, I will do all those things, sir.
Okay, no problem. Yeah, I mean, it's good that you are attending the courses and you are interested. We really look forward that you find a water body yeah.
Already I found a water body and I took some samples also. Due to holidays and some personal reasons, I couldn't did those projects.
No worries, no worries. Yeah, let us know when if you need help, and yeah.
Okay.
Let's see if we have anyone else. So Inian and A. Santosh are still fixing their audio, I'm assuming. Whenever you are ready, you can speak up. Let me see what's next. Okay, the form that we have shared, we urge you to fill that up. It also has an option to add the timeline of the project, like when do you want to start it and how do you want to start it and all those things. So that would be great to know. So that way we can help you in a much more efficiently. And I talked a little bit about the kind of documentation that we plan to do. So once we start having data, like all of us, together, because remember, we all are in a big, huge project and we all want to do something with the data and make sense of the data. So we'll take responsibility of organizing the data once we have enough data. And then we'll see how we want to publish it. Definitely we'll have to create an AdGeo report. And then on top of that, if the data makes sense or some of us see something very interesting, we'll try to organize the publication as efficiently as possible. Is there anything else that I'm missing? There is going to be a guest lecture series that Kanika is going to lead us in. What we plan to do is we can have these monthly meetups, right? So based on your convenience and whenever, we do not have to meet all of us at the same time. That is the tutorial part. But for the guest lecture, what we are trying to do is we are inviting people citizen scientists or professional scientists who are working in the domain of algae, both marine and freshwater. And they will give us some more knowledge, information wise, and also their experience traveling the world and doing sampling. So we will update you on that. And if Kanika wants to say a word or two about how that would look like.
Yeah, so the idea is, we want to stay connected, and at the same time, we want to think about how can we expand this project as well, so that we can reach different parts of the world and different kinds of lakes and everything, because, like this is a pilot project. We want to see where we can go from there. So for keeping people engaged, like the idea is we'll invite, maybe scientists who are working on different aspects of algae, and also, as Iqbal mentioned, citizen scientists who have been doing this kind of sampling, maybe for algae or other kind of water bodies and things like that. So we want to have those breadth of people, and it will probably be most likely once a month something. And for that, actually, I would request if, because it's going to be online, so I would actually like if all your students can also join the link. So it's not only for teachers, but also we want to reach out to as many students as possible. And it's going to be a 1 hour thing, so maybe 30 minutes of lecture and 30 minutes of questions and talking time, because we really want these to be sort of an interactive seminars. They will probably tell about what kind of work or research they're doing in the field or in the lab, and then we can ask them questions which can be about our sampling. And the thing is, we also want to get kids excited about the marine life in general and everything. So for those, because it's going to be online, sort of a more series like thing, it'll be really great if, whenever we forward you the link, if you can send it to your classes and students, whoever can join. It's going to be open to everybody, so whoever can join, if that'll be really great, I think. Yeah.
So
I think,
yeah, go ahead, Ankit.
I think even the parents can join, right?
Oh yes, definitely, anybody. So it's going to be open to anybody in the world. Yeah.
And I think the cohort that we have right now and the locations is fantastic, and we would try our best to supplement with additional materials if you want to expand your student cohort or add your friends and you need more material in the form of tools, Foldscope and Spectroscope. So we try to supplement that. And we are also open to expanding cohort beyond your geographical location. So that way, but whatever we have, please feel free to use the equipment that we have given you and not be conservative about it. Use it as much as you want. Even if you break one, we'll try to supplement as soon as possible.
Yeah, and sharing is caring. So, you feel free to share with other people who, other teachers, students, whoever. So we really want to expand this program going forward, and we are trying our best, and we want to, maybe start some satellite chapters going in the future, so that like in India or in other parts of the world. So yeah, do that. Thank you.
All right. Yeah, that's about it. Do we have anything else, Ankita?
Maybe we can talk about how frequently we are going to meet, because that's something we also wanted feedback from you all. We were thinking maybe twice a month or once a month, but please let us know. I mean, I just want to open this discussion, and what frequency do you feel like you can always email us, as we said, and I'm just emphasizing it again and again, because just drop us an email if you feel like you are stuck somewhere, or maybe you missed a month and you feel like, oh my God, this is missing something. Don't worry about all those things. Like I know life happens to all of us, so if you miss a month or something, don't worry too much about it. Please continue. Missing a data point is not the end of the world. What we want really is to you feel excited, your students feel excited about it, and increase curiosity.
Those of you who have not sampled yet and you are waiting to go to the water body, my recommendation would be to sample anything that you find. Finding that one date and going to the water body. You can do onion peels, you can do paper, your own hairs, anything that you find. And I promise it's going to be really exciting, and it will give you a head start into the project.
And I think this is going to be especially useful for people who are first-time users of Foldscope. Just keep exploring. I think that's how you will get better at it. And there is also, we have sent links previously to Microcosmos website, so you can learn some tricks and techniques. People who have been doing this for years, for samples like onion peels or things like that, or even water, like what are some, tips and tricks to get good pictures and everything. So the more you practice with it, I think in the next month or so, the better you'll get. And like, you never know what interesting things you might find, because I remember like Manu Prakash, he had posted sometime back that they in Pune somewhere, I think, like they discovered a new species of algae using Foldscope. So you might discover something new which we don't know.
Yeah, I mean, he identified also oscillatoria, and he was telling us, because the gliding thing I showed,
Yeah.
that they're going to come up with a paper now to, like they worked 5 years after
Right.
discovering that on
Yeah, I
Foldscope.
mean, this is a lesson that I learned a little later. So, I mean, we all lose the childlike curiosity as we grow up. So don't take things for granted. Like something that you see that may be a little insignificant, a little from a drop of water in the pond, but it has a lot of scientific value. So people are using the tools that we have given you and actually writing scientific papers and making discoveries all over the world. So yeah. All right.
Iqbal, Dr. Vasavi wants to say.
Yes.
Slides from our laboratory using Foldscope, but it didn't work out properly. Yeah.
Actually, I can't hear you.
Dr. Vasavi, I think the laptop or the computer microphone that you're using, that is working, not the one that you have close to your mouth. So that's why we can't hear you properly.
Did we
Now,
lose her?
yeah, now we don't see you as well.
The thing I heard she was saying about
Maybe you can type your question. Meanwhile, we can address other questions, right? Then we can answer. There's another person who had issues with account from it's Nehalata, I think.
Maybe we can start wrapping up. And
Yeah,
somebody,
I mean, we are almost done. Yeah, if anyone doesn't have any question, people doesn't have any question, then yeah, feel free, enjoy your weekend. We'll inform you about our next meetup. Most likely we'll try to do twice a month initially, and we'll tune it down a little bit as people get much more mature in using the equipment.
And like, if there's any questions regarding upload or anything like that, just feel free to email us whatever question you have.
Yeah.
And we'll respond back.
Yeah.
So Dr. Vasavi said she tried some of the permanent slides from the lab and it's not clear with the Foldscope. I mean, if you want, we can sit with you as well. But one of the few things which often is an issue when focusing with Foldscope is to make sure that you're on the right side with respect to the sample, like you're not viewing it from the side of the slide, but the microscope slide, but more towards the cover slip. So the cover slip should be closer to the lens.
Maybe Iqbal, can you show with the Foldscope, like where the slide should be?
Okay, so she's saying that was placed properly. The second thing often is with the type of the phone. So yeah, like some of us struggle based on how far it is from our phone camera.
Right.
Are you able to see it with your eye, Dr. Vasavi? Because that gives an idea of whether it's the phone issue. Okay, so I will really suggest first trying it with your eye towards like some light source, because then you can move push the slide closer or a little far and see if you're able to see that, because then you would know whether it's the problem with the placement of the slide or it's with the phone the issue is.
and I've also found that if I keep like my Foldscope on some sort of an elevated object, like a book or something, and then I try to adjust the focus, it's always a little easier that way. So there should be like some height, because so the focus can be properly, but maybe you can also just send us pictures. So like what exactly it doesn't look like, so we can think more about it. I thought.
Yeah. I mean, one thing that I can do quickly is if someone can pin my screen, then what I'll do is I can show you. Yeah, I can show you how the slide looks like. So as Ankita mentioned, that the slide has a cover slip on top. So you try to align this cover slip towards the towards the lens. Okay, so let me try to hold it first. Okay, so you open the flap like that. All right, like that, and align the cover slip towards the lens. Okay, so then you have to find a way to insert the cover slip through the paper cutting there and insert it like that, right? And then you flip. Also, manually, you can align the sample with the lens. Let me see. Yeah, something like that. So this way you'll get much more accuracy in terms of visualizing what you want to see. First thing would be to visualize it with your eyes like that. Okay, and point it towards a light source and then try to see what you are looking at. And also it takes some time to sort of use your fingers and then moving it up and down and back and forth like that. Okay.
And just to add on, sorry, Iqbal, if you don't feel like the light source is good enough, there's always that small LED thing which you can use.
Right. So the LED stuff that there's a LED source, which is this one. You know that you have to take this piece of plastic out if you want to engage the battery and turn it on, and then you just take it closer to the back side of the horoscope and stick it there. It automatically aligns, so you don't have to worry about anything. So now you have your own light source, even if you are in a darker room. And again, visualize. Okay. Once you know that you are on top of the sample and you can visualize by eye, then think about connecting the cell phone to that, right? And in order to do that, you have to think about, if you have multiple cameras on your cell phone, right? You have to find out which one is the right camera. Then the adapter has to stick in there. And also you have to make sure the alignment is correct, because the adapter here is going to fit in with that one. So they are magnets, right? So if the alignment is not right, you will automatically find out, because they will repel one another, right? So these things, just tinker around and find you. It's quite intuitive in my mind. But if you still have issues, we can have a separate a tutorial session only for you. Let us know.
Yeah, also like regarding the website accounts, I thought someone was saying that they were getting incorrect username and password. Make sure if you don't have an account yet, you have to click on the sign up button, not the login button. Make sure you're doing that, and then you'll be able to get an account first. That way, and then you can log in with that username password.
Yeah, I think that maybe we can create a document with snapshots of where to click and where not to click, and then we can sort of give that document like
okay. Yeah, it's in the earlier video, but yeah, okay, we can create a document. Yeah.
We can also send emails to people who we haven't received the login or sign up information from, sorry. And then, yeah, we will let you know that we haven't received your sign up.
Right.
Cool.
All right.
Any more questions? Thank you.
Thank you so much, everyone. And thanks for bearing with us in the last 1 month. I think we had a fantastic workshop, and claps for everyone. Okay. So,
thank you.
We really enjoyed learning with you, and we learned a lot actually in the process. And we really hope that you stick with us and stay on the project and find something interesting for the sake of science and for the sake of curiosity.